Welcome to a brand new explainer. I am so thrilled you're here today because we're about to completely change the way you look at your kitchen. When you step in there to cook, you probably just see pots, pans, and pantry staples. But if we put on our scientist goggles and focus entirely on the science for a moment, that room is actually a high-tech laboratory. Literally every time you bake, cook, or even just slice an apple, you're conducting fascinating, complex experiments in chemistry and physics. So today we are going to uncover the invisible scientific marvels hiding in the food you eat every single day. Let's just dive right in.
Here is our tasting menu for today's explainer. First, the power of pH, then the molecules and instant pudding, followed by the Maillard reaction, emulsions and butter physics, enzymes and dehydration, and finally, we'll look at chemical versus physical changes. All right, let's kick things off with section 1, the power of pH.
To really grasp food science, we've got to start at the molecular baseline with pH. Now, pH literally stands for the potential of hydrogen. It's a logarithmic scale running from 0 to 14 that measures exactly how acidic or alkaline a solution is. A pH of exactly 7 is neutral. Anything below 7 is acidic, like lemon juice or vinegar, and anything above 7 is alkaline. Understanding this scale is absolutely critical because manipulating this one single variable completely alters the food we eat. The potential of hydrogen dictates a surprising number of things in our food. It essentially acts as a master switch for enzyme control, flavor enhancement, and texture modification. And because dangerous pathogens absolutely hate acidic environments, lowering the pH provides crucial microbial stability to prevent spoilage. It's built-in preservation. Let's look at how this actually plays out. When you lower the pH of milk, it causes the casein proteins to denature. Basically, their structures unfold and clump together, locking water and fat into a gel. And boom, that is the exact science of making cheese. Or if you expose starch to an acid, it triggers hydrolysis, breaking down long starch chains to create the amazingly smooth texture of something like lemon curd. pH even dictates how emulsifiers hold oil and water together in mayonnaise by altering their electrical charge. And get this, when it comes to taste, acidic environments can literally mask sweetness by protonating the taste receptors on your tongue. So a shift in pH doesn't just change the flavor, it physically changes the shape and behavior of the food's molecules. Speaking of altering molecular shapes, let's move to section 2, molecules in instant pudding. Traditional pudding requires heat to force starch granules to swell up and thicken, but instant pudding sets cold in just a few minutes. How is that possible? Well, the secret is this brilliant piece of 1950s food technology called pre-gelatinized starch. Scientists basically pre-cook the starch, which totally disrupts its tightly packed crystalline structure, and then they dry it out. This turns those starch granules into microscopic sponges that are primed and ready to rapidly absorb liquid and swell up even at room temperature. But here's a really fascinating question. Why does the box strictly tell you to use cold milk instead of water? I mean, if you whisk that pre-gelatinized starch with water, it definitely thickens, but it's going to have a super weak body and a weirdly chalky mouthfeel. When you use milk, you're introducing complex biological components, specifically casein and whey proteins, along with milk fats. Those proteins chemically interact with the starch network to give it a much richer structure, while the milk fats coat your tongue, enhancing that creamy perception and completely suppressing those gross, chalky notes from the starch. So when you're mixing instant pudding, you're actually triggering a highly choreographed chemical ballet. The pre-gelatinized starch rapidly pulls in water and swells into this massive gel network. But to stop that network from just turning into a lumpy, unappetizing mess, powerful emulsifiers like tetrasodium pyrophosphate go to work, keeping all those fat molecules evenly dispersed. The sugar dissolves to balance out the texture, and as it chills in the fridge, the entire molecular network locks firmly into place. It's just a beautifully engineered cold-setting reaction. From cold-setting reactions, let's turn up the heat in section 3, the Maillard reaction. If there is only one chemical reaction you memorize from this entire explainer, it's gotta be this one. First described by a chemist named Louis-Camille Maillard back in 1912, the Maillard reaction is a non-enzymatic Browning reaction. It happens when amino acids, which come from proteins, violently collide and react with reducing sugars. But this reaction has a strict kinetic energy threshold. It generally only kicks off at temperatures above 140 degrees Celsius, or about 284 degrees Fahrenheit. This chemical reaction doesn't just make your food look brown, it synthesizes completely new molecular structures called melanoidins, which act as brown pigments. And at the exact same time, it generates hundreds of distinct, highly volatile aroma and flavor compounds. Whether it's the beautiful crust on baked bread, the hard sear in a steak, or those incredible roasted notes in your morning coffee, you are quite literally tasting the complex chemical byproducts of the Maillard reaction. Actually, let's clear up a massive misconception right now. A lot of people think that just making something hot and oily automatically causes it to brown. No way. Pure fat alone will never, ever undergo the Maillard reaction. The science is absolute on this. You must have a molecular collision between a protein source and a reducing sugar. Without both of those components present, the rich, complex flavors of the Maillard reaction simply cannot form. So we've looked at chemical heat, but what about physical agitation? Let's get into section 4, emulsions and butter physics. Heavy cream is honestly a total marvel of fluid dynamics. It's an emulsion, meaning it's a mixture of two liquids that naturally hate each other and refuse to stay mixed, oil and water. In cream, millions of tiny fat droplets are suspended and dispersed all throughout the watery part of the milk. Now, if you've ever tried to make butter by shaking cream in a mason jar, you know it's a serious arm workout. But temperature plays a huge role in the physics here. If you use room temperature cream, the molecules have much higher kinetic energy. They're vibrating and zipping around much faster, which allows the fat globules to smash into each other and clump together super quickly. But if that cream is chilled, the molecules have lower kinetic energy. They move sluggishly, and forcing them to form butter takes significantly longer. So what's the exact physics inside that shaking jar? Those tiny fat globules in the cream are surrounded by delicate protective membranes made of phospholipids and proteins. When you agitate the cream violently, you physically damage and strip away those protective membranes. The newly freed fat molecules instantly seek each other out and conjoin. And then a massive phase shift happens. The cream, which started as an oil and water emulsion, literally inverts. It becomes a water and oil emulsion, which we call butter, and all that remaining liquid simply separates out as buttermilk. It's a violent, purely physical transformation happening right on your kitchen counter. Let's move on to the science of food preservation in section 5, enzymes and dehydration. You know that really frustrating thing where you slice an apple and it almost immediately turns brown? That's not just oxygen. It's a very specific chemical event called enzymatic browning. When your knife slices the apple, it damages the cell walls. This lets oxygen from the air rush in and mix with the apple's natural phenolic compounds. But the real culprit is a catalyst inside the apple called PPO, or polyphenol oxidase. This enzyme basically hits the gas pedal on the whole reaction, rapidly churning out brown-colored pigments called melanins. So how do we use science to fight back and defeat this enzyme? Well, we've got two primary weapons. We can lower the pH by hitting the apple with ascorbic acid, like a squeeze of lemon juice. This highly acidic environment severely cripples the PPO enzyme's activity. Or we go with the nuclear option, heat. By blanching the fruit in boiling water, we apply thermal energy to physically warp the shape of the enzyme, which is a process called denaturation. Once its shape is ruined, the enzyme is permanently broken, and browning just stops completely. So we can halt enzymes with acid or heat, but another amazing method of preservation is dehydration. By extracting moisture, we make the food mathematically hostile to microbes. They simply die or go dormant without water. Think about a fresh apple slice weighing 50 grams. If you dehydrate it down to a 10-gram chip, you've removed 40 grams of pure water mass. That means that fresh apple slice was exactly 80% water before you started. It really puts into perspective just how much of the food we eat is basically just structurally supported water. For our final section, let's draw a really distinct scientific boundary between two completely different types of reactions. Section 6, chemical and physical changes. To really see the difference, let's compare a microwave mug cake to cotton candy. When you bake that mug cake, you rely on chemical leavening, usually from baking powder. Baking powder has sodium bicarbonate and acid salts. When you mix them with liquid and heat them up, a brand new chemical reaction occurs, literally synthesizing an entirely new substance, carbon dioxide gas. Those CO2 bubbles expand and leaven the cake. That is a textbook chemical change. Cotton candy, on the other hand, might look like pure magic, but it is a strictly physical change. Sucrose, or sugar, is melted into a liquid state and spun at incredibly high speeds. Centrifugal force pushes that liquid sugar through tiny holes, stretching it into microscopic threads that instantly cool in the air. The molecular structure of the sugar hasn't changed one bit. No new chemical was created. It just changed physical states from a crystal to a liquid to glass-like solid strands. So from protonating your taste receptors with pH to the physical inversion of butter emulsions, right down to the Maillard reaction synthesizing hundreds of complex flavors through heat, your kitchen is undeniably a high-tech laboratory of science. The next time you step up to your stove or even just slice into an apple, I want you to look just a little bit closer. What other invisible chemical reactions and physical changes are you eating every single day? Stay curious, keep experimenting, and we'll see ya in the next explainer.